Chapter 9 Biomolecules. There is a wide diversity in living organisms in our biosphere. Now, a question that arises in our mind is: are all living organisms made up of the same chemicals? That is, elements and compounds. You have learned in chemistry how elemental analysis is performed. If we perform such an analysis on a plant tissue, animal tissue, or a microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, and several others in their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of earth crust as an example of non living matter, we obtain a similar list. What are the differences between the two lists? In absolute terms, no such differences could be made out. All the elements present in a sample of earth crust are also present in a sample of living tissue. However, a closer examination revealed that the relative abundance of carbon and hydrogen with respect to other elements is higher in a living organism than in earth crust. 9.1 How to analyze chemical composition. We can continue asking in the same way: what type of organic compounds are found in living organisms? How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue, a vegetable or a piece of liver, etc., and grind it in trichloroacetic acid Cl3 CCOOH using a mortar and a pestle. We obtain a thick slurry. If we were to strain this through a cheesecloth or cotton, we would obtain two fractions. One is called the filtrate, or more technically, the acid soluble pool, and the second, the retentate, or the acid insoluble fraction. Scientists have found thousands of organic compounds in the acid soluble pool. In higher classes, you will learn about how to analyze a living tissue sample and identify a particular organic compound. It will suffice to say here that one extracts the compound, then subjects the extracts to various separation techniques till one has separated a compound from all other compounds. In other words, one isolates and purifies a compound. Analytical techniques, when applied to compound, give us an idea of the molecular formula and the probable structure of the compound. All the carbon compounds that we get from living tissues can be called biomolecules. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive experiment has to be done. One way is a simple amount of living tissue, say a leaf or a liver, and this is called wet weight, and dry it. All the water evaporates. The remaining material gives dry weight. Now, if the tissue is burned fully, all the compounds, carbon, are oxidized to gaseous forms like CO2, water vapor, and are removed. What is remaining is called ash. This ash containing inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in this acid soluble fraction. Therefore, elemental analysis gives form of hydrogen, oxygen, chlorine, carbon, etc., while analysis from compounds give an idea of the kind of organic and inorganic constituents present in living tissues. From a chemistry point of view, one can identify functional groups like aldehydes, ketones, aromatic compounds, etc. But from a biological point of view, we shall classify them into amino acids, nucleotide, bases, fatty acids, etc. Amino acids are organic compounds containing an amino group and an acidic group as substituents on the same carbon. That is the alpha carbon. Hence, they are called alpha amino acids. They are substituted methanes. There are four substituent groups occupying the four valency positions. These are hydrogen carboxyl group, amino group, and variable group, designated as R group. Based on the nature of R group, there are many amino acids. However, those which occur in proteins are only of 20 types. The R group in these proteinaceous amino acids could be a hydrogen. The amino acid is called glycine, a methyl group alanine, hydroxyl methyl serine, etc. Three of the 20 are shown in figure 9.1. The chemical and physical properties of amino acids are essentially of the amino, carboxyl, and the R functional groups. Based on the number of amino and carboxyl groups, there are acidic, example glutamic acid, basic lysine, and neutral valine amino acids. Similarly, there are aromatic amino acids, triosine, phenylalanine tryptophan. A particular property of amino acids is the ionizable nature of NH2 and COOH group. Hence, in solutions of different pH, the structure of amino acid changes. Lipids are generally water insoluble. They could be simple fatty acids. A fatty acid has a carboxyl group attached to an R group. The R group could be methyl or ethyl or a high number of CH2 group, 1 carbon to 19 carbons. For example, palmitic acid has 16 carbons, including carboxyl carbon. Arachidonic acid has 20 carbon atoms, including the carboxyl carbon. Fatty acids could be saturated without double bond or unsaturated with one or more carbon-carbon double bonds. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Here, the fatty acids are found esterified with glycerol. They can be then monoglycerides, diglycerides, and triglycerides. These are also called fats and oil based on melting points. Oils have lower melting point, example gingerly oil, and hence remain as oil in winters. Can you identify a fat from the market? Some lipids have phosphorus and phosphorylated organic compound in them. These are phospholipids. They are found in cell membrane. Lecithin is one example. Some tissues, especially the neural tissues, have lipids with more complex structures. Living organisms have a number of carbon compounds in which heterocyclic rings can be found. Some of these are nitrogen bases: adenine, guanine, cytosine, uracil, and thymine. When found attached to a sugar, these are called nucleosides. If a phosphate group is also found esterified to the sugar, they are called nucleotides. Adenosine, guanosine, thymidine, uridine, and cytidine are nucleosides. Adenylic acid, thymidyllic acid, guanylic acid, uridylic acid, and cytidyllic acids are nucleotides. Nucleic acids are like DNA and RNA consist of nucleotides only. DNA and RNA function as genetic material. 9.2 Primary and secondary metabolites. The most exciting aspect of chemistry deals with isolating thousands of compounds, small and big, from living organisms, determining their structure and, if possible, synthesizing them. If one were to make a list of biomolecules, such a list would have thousands of organic compounds, including amino acids, sugar, etc. For reasons that are given in section 9.10, we can call these biomolecules as metabolites. In animal tissues, one notices the presence of all such categories of compounds shown in figure 9.1. These are called primary metabolites. However, when one analyzes plant, fungal, and microbial cell, one would see thousands of compounds other than these primary metabolites. Example: alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gum, spices, etc. These are called secondary metabolites. While primary metabolites have identifiable function and play known roles in normal physiological processes, we do not at the moment understand the role or function of all the secondary metabolites in host organisms. However, many of them are useful to human welfare, example rubber, drugs, spices, scents, and pigments. Some secondary metabolites have ecological importance. In the later chapters and years, you will learn more about this. 9.3 Biomacromolecules. There is one feature common to all those compounds found in the acid soluble pool. They have molecular weights ranging from 18 to around 800 daltons approximately. The acid insoluble fraction has only four types of organic compounds, that is proteins, nucleic acid, polysaccharides, and lipids. These classes of compounds, with the exception of lipids, have molecular weights in the range of 10,000 daltons and above. For this very reason, biomolecules, that is chemical compounds found in living organisms, are of two types. One, those which have molecular weights less than 1000 dalton and are usually referred to as micromolecules or simply biomolecules, while those which are found in the acid insoluble fraction are called macromolecules or biomacromolecules. The molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. Then why do lipids whose molecular weights do not exceed 800 Dalton come under acid insoluble fraction, that is macromolecular fraction? Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structure like cell membrane and other membranes. When we grind a tissue, we are disrupting the cell structure. Cell membrane and other membranes are broken into pieces and form vesicles which are not water soluble. Therefore, these membrane fragments in the form of vesicles get separated along with the acid insoluble pool and hence in the macromolecular fraction. Lipids are not strictly macromolecules. The acid soluble pool represents roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid insoluble fraction. Together, they represent the entire chemical composition of living tissues or organisms. In summary, if we represent the chemical composition of living tissues from abundance point of view and arrange them class-wise, we observe that water is the most abundant chemical in living organisms. 9.4 Proteins. Proteins are polypeptides. They are linear chains of amino acids linked by peptide bonds as shown in figure 9.3. Each protein is a polymer of amino acids. As there are 20 types of amino acids, example alanine, cytine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer repeating n number of times. This information about the amino acids is more important as later in our nutrition lessons you will learn that certain amino acids are essential for our health and they have to be supplied throughout our diet. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non-essential. The latter are those which our body can make while we get essential amino acids through our diet or food. Proteins carry out many functions in living organisms. Some transport nutrients across cell membranes, some fight infectious organisms, some are hormones, some are enzymes, etc. Collagen is the most abundant protein in animal world and ribulose biphosphate carboxylase oxygenates, that is Rubisco, is the most abundant protein in the whole of the biosphere. 9.5 Polysaccharides. The acid insoluble pellet also has polysaccharides carbohydrate as another class of macromolecules. Polysaccharides are long chains of sugars. They are threads, literally a cotton thread, containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. Cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of energy in plant tissues. Animals have another variant called glycogen, insulin. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. Cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of energy in plant tissues. Animals have another variant called glycogen. Inulin is a polymer of fructose in a polysaccharide chain, say glycogen. The right end is called the reducing end and the left end is called the non-reducing end. It has branches as shown in the form of the cartoon figure 9.2. Starch form helical secondary structures. In fact, starch can hold iodine molecules in the helical portion. The starch iodine is blue in color. Cellulose does not contain complex helices and hence cannot hold iodine. Plant cell walls are made up of cellulose. Plant made from plant pulp and cotton fiber is celluloses. There are more complex polysaccharides in nature. They have a building blocks, amino sugars, and chemically modified sugars, example glucosamine, N-acetylglucotamine, etc., galactosamine, etc. Exoskeletons of arthropods, for example, have a complex polysaccharides called chitin. These complex polysaccharides are mostly homopolymers. 9.6 Nucleic acids. Another type of macromolecule that one would find in the acid insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides. Together with polysaccharides and polypeptides, these comprise the true macromolecular function of any living tissue or cell. For nucleic acid, the building block is a nucleotide. A nucleotide has three chemically distinct components. One is a heterocyclic compound, the second is a monosaccharide, and the third is phosphoric acid or phosphate. As you notice in figure 9.1, the heterocyclic compounds in nucleic acid are the nitrogenous bases, namely adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are substituted purines, while the rest are substituted pyrimidines. The skeletal heterocyclic ring is called a purine and the pyrimidine, respectively. The sugar found in polynucleotides is either ribose, a monosaccharide pentose, or secondary deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid, that is DNA, while which contains ribose is called ribonucleic acid, RNA. 9.7 Structure of proteins. Proteins, as mentioned earlier, are heteropolymers containing strings of amino acids. Structure of molecules means different things in different contexts. In inorganic chemistry, the structure invariably refers to the molecular formula, example NaCl, MgCl2, etc. Organic chemists always write a two-dimensional view of molecules while representing the structure of the molecule, example benzene, naphthalene, etc. Physicists conjure up the three-dimensional views of molecular structure while biologists describe the protein structure at four levels. The sequence of amino acid, that is the positional information in a protein, which is the first amino acid, which is the second, and so on, is called the primary structure. Figure 9.3a of a protein. A protein is imagined as a line, the left end represented by the first amino acid and the right end represented by the last amino acid. The first amino acid is also called the N-terminal amino acid. The last amino acid is called the C-terminal amino acid. A protein thread does not exist throughout an extended rigid rod. Other regions of the protein thread are folded into other forms in which is called the secondary structure. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to a tertiary structure. This gives us a three-dimensional view of a protein. Tertiary structure is absolutely necessary for the many biological activities of protein. Some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individual folded polypeptides or subunits are arranged with respect to each other, example linear string of spheres arranged once upon each other in the form of a cube or a plate, etc., is the architecture of a protein, otherwise called quaternary structure of a protein. Adult human hemoglobin consists of four subunits. Two of these are identical to each other. Hence, two subunits of alpha type and two subunits of beta type together constitute the human hemoglobin. 9.8 Nature of bond linking monomers in a polymer. In a polypeptide or protein, amino acids are linked by a peptide bond which is formed by when the carboxyl COOH group of one amino acid reacts with the amino NH2 group of next amino acid. With the elimination of a water moiety, the process is called dehydration. In a polysaccharide, the individual monosaccharides are linked by a glycosidic bond. This bond is also formed by dehydration. This bond is formed between two carbon atoms of two adjacent monosaccharides. In a nucleic acid, a phosphate moiety links 3 carbon of one sugar of one nucleotide to the 5 carbon of the sugar of the succeeding nucleotide. The bond between the phosphate and the hydroxyl group of sugar is an ester bond. As there is one such ester bond on either side, it is called phosphodiester bond. Nucleic acid exhibit a wide variety of secondary structure. For example, one of the secondary structure exhibited by DNA is the famous Watson-Crick model. This model says that the DNA exists as a double helix. The two strands of the polynucleotides are antiparallel, that is, run in the opposite direction. The backbone is formed by the sugar-phosphate-sugar chain. The nitrogen bases are projected more or less perpendicular to this backbone but face inside. A and G of one strand compulsorily base pairs with T and C, respectively, on the other strand. There are 2 hydrogen bonds between A and T and 3 hydrogen bonds between G and C. Each strands appear like a helical staircase. Each step of ascent is represented by a pair of bases. At each step of the ascent, the strand turns 36 knot. One full turn of helical strand would involve 10 steps or 10 base pairs. Attempt drawing a line diagram. The pitch would be 34 knot. The rest per base pair would be 3.4 Armstrong. This form of DNA with the above mentioned silent features is called bDNA. In higher classes, you will be told that there are more than a dozen forms of DNA named after English alphabets with unique structural features. 9.9 Dynamic state of body constituents concept of metabolism. What we have learned till now is that living organisms, be it simple bacterial cell, a protozoan, a plant, or an animal, contain thousands of organic compounds. These compounds or biomolecules are present in cell concentration expressed as moles per cell or moles per liter, etc. One of the greatest discoveries ever made was the observation that all the biomolecules have a turnover. This means that they are constantly being changed into some other biomolecules and also made from some other biomolecules. This breaking and making is through chemical reaction constantly occurring in a living organisms. Together, all these chemical reactions are called metabolism. Each of the metabolic reactions result in the transformation of biomolecules. A few examples for such metabolic transformations are removal of carbon dioxide from amino acids, making an amino acids into an amine, removal of amino group in a nucleotide base, hydrolysis of glycosidic bond in disaccharides, etc. We can list tens and thousands of such examples. Majority of these metabolic reactions do not occur in isolation but are always linked to some other reactions. In other words, metabolites are converted into each other in a series of linked reactions called metabolic pathways. These metabolic pathways are similar to the automobile traffic in a city. These pathways are either linear or circular. These pathways crisscross each other, that is, there are traffic junction. Flow of metabolites through metabolic pathway has a definite rate and direction like automobile traffic. This metabolite flow is called the dynamic state of body constituents. What is most important is that this interlinked metabolic traffic is very smooth and without a single reported mishap for healthy condition. Another feature of this metabolic reaction is that every chemical reaction is catalyzed reaction. There is no uncatalyzed metabolic conversion in a living system. Even CO2 dissolving in water, a physical process, is a catalyzed reaction in living system. The catalyst which hasten the rate of a given metabolic conversion are also protein. These proteins with catalytic power are named enzymes. 9.10 Metabolic basis for living. Metabolic pathways can lead to more complex structure from a simpler structure. For example, acetic acid become cholesterol or lead to a simple structure from a complex structure. For example, glucose becomes lactic acid in a skeletal muscle. The former cases are called biosynthetic pathways or anabolic pathways. The later constitute degradation and hence are called catabolic pathways. Anabolic pathways, as expected, consume energy. Assembly of a protein from amino acids require energy input. On the other hand, catabolic pathways lead to the release of energy. For example, when glucose is degraded to lactic acid in a skeletal muscle, energy is liberated. This metabolic pathway from glucose to lactic acid, which occurs in 10 metabolic steps, is called glycolysis. Living organisms have learned to trap this energy liberated during degradation and store it in the form of chemical bonds. As and when needed, this bond energy is utilized for biosynthetic, osmotic, and mechanical work that we perform. The most important form of energy currency in living system is the bond energy in chemical called adenosine triphosphate ATP. How do living organisms derive their energy? What strategies have they evolved? How do we store this energy and in what form? How do they convert this energy into work? You will study and understand all this under a subdiscipline called bioenergetics later in your later higher classes. 9.11 The living state. At this level, you must understand that the tens and the thousands of chemical compounds in a living organism, otherwise called metabolites or biomolecules, are present at concentration characteristic of each of them. For example, the blood concentration of glucose in a normal healthy individual is 4.2 mole per liter to 6.1 mole per liter. while that of hormones would be nanograms per milliliter. The most important fact of biological system is that all living organisms exist in a steady state characterized by concentration of each of these biomolecules. These biomolecules are in a metabolic flux. Any chemical or physical process moves spontaneously to equilibrium. The steady state is non-equilibrium state. One should remember from physics that systems at equilibrium cannot perform work. As living organisms work continuously, they cannot afford to reach equilibrium. Hence, the living state is a non-equilibrium steady state to able to perform work. Living organism is a constant effort to prevent falling into equilibrium. This is achieved by energy input. Metabolism provides a mechanism for the production of energy. Hence, the living state and metabolism are synonymous. Without metabolic, there cannot be a living state. 9.12 Enzyme. Almost all enzymes are proteins. There are some nucleic acid that behaves like enzymes. These are called ribozymes. One can depict an enzyme by a line diagram. An enzyme like a protein has a primary structure, that is, amino acid sequence of the protein. An enzyme like a protein has the secondary and tertiary structure. When you look at a tertiary structure, you will notice that the backbone of the protein chain folds upon itself. The chain crisscrosses itself, and hence many crevices or pockets are made. One such pocket is the active site. An active site of an enzyme is a crevice or pocket into which the substrate fits. This enzyme through the active site catalyzed reaction at a high rate. Enzyme catalysis differ from inorganic catalysts in many ways, but one major difference need mention. Inorganic catalysts work efficiently at high temperature and high pressure, while enzymes get damaged at high temperature, say above 40 degrees Celsius. However, enzymes isolated from organisms who normally live under extremely high temperatures, example hot vents and sulfur springs, are stable and return their catalytic power even at high temperatures up to 80 to 90 degrees Celsius. Thermal stability is thus an important quality of such enzyme isolated from thermophilic organisms. 9.12.1 Chemical reactions. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compounds undergo two types of changes. A physical change simply refers to a change in shape without breaking of bonds. This is called a physical process. Another physical process is change in state of matter when ice melts into water or when water becomes a vapor. These are also physical processes. However, when bonds are broken and new bonds are formed during transformation, this is called a chemical reaction. For example, BaOH2 plus H2SO4 will give BaSO4 plus 2H2O is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. Rate of physical or chemical processes refers to the amount of product performed per unit time. It can be expressed as rate is equal to delta product upon delta time. Rate can also be called velocity if the direction is specified. Rates of physical and chemical processes are influenced by temperatures among other factors. A general thumb rule is that rate doubles or decreases by half for every 10 degrees Celsius change in either direction. Catalyzed reactions proceed at rates vastly higher than that of uncatalyzed ones. When enzyme catalyzed reactions are observed, the rate would be vastly higher than the same but uncatalyzed reaction. For example, carbon dioxide plus water will give carbonic acid with the help of enzyme carbonic anhydrase. In the absence of any enzyme, this reaction is very slow, with about 200 molecules of H2CO3 being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 6,000 molecules being formed every second. The enzyme has accelerated the reaction rate about 10 million times. The power of enzymes is incredible indeed. There are thousands of types of enzyme, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction, when each of the step is catalyzed, by the same enzyme complex or different enzyme is called metabolic pathway. For example, glucose gives 2 pyruvic acid is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzyme catalyzed metabolic reactions. When you study in respiration in chapter number 14, you will study these reactions. At this stage, you should know that this very metabolic pathway with one or two additional reaction give rise to a variety of metabolic end products. In our skeletal muscle under anaerobic condition, lactic acid is formed. Under normal aerobic condition, pyruvic acid is formed. In yeast, during fermentation, the same pathway leads to the production of ethanol, that is alcohol. Hence, in different condition, different products are possible. 9.12.2 How do enzymes bring about such high rates of chemical conversion? To understand this, we should study enzymes a little more. We have already understood the idea of an active site. The chemical or metabolic conversion refers to a reaction. The chemical which is converted into a product is called the substrate. Hence, enzyme, that is proteins with three-dimensional structures, including an active site, convert a substrate into a product. Symbolically, this can be depicted as S is converted to P. It is now understood that the substrate S has to bind the enzyme at its active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation of ES complex. E stands for enzyme. This complex formation is a transient phenomenon. During the state where substrate is bound to the enzyme active site, a new structure of the substrate called transition state structure is formed. Very soon after the expected bond breaking making is completed, the product is released from the active site. In other words, the structure of substrate gets transformed into the structure of products. The pathway of this transformation must go through the so-called transition state structure. There could be many more altered structural states between the stable substrate and the product. Implicit in this statement is the fact that all the other intermediate structural states are unstable. Stability is something related to the energy status of the molecule or the structure. Hence, when we look at this pictorially through a graph, it looks like something as in figure 9.6. The y-axis represents the potential energy content. The x-axis represents the progression of the structural transformation or states through the transition state. You would not you would notice two things. The energy level difference between S and P. If P is at the lower level than S, the reaction is exothermic reaction. One need not supply energy by heating in order to form the product. However, whether it is an exothermic or spontaneous reaction or an endothermic or energy requiring reaction, the S has to go through a much higher energy state or transition state. The difference in average energy content of S from that of the transition state is called activation energy. Enzymes eventually bring down this energy barrier, making the transition of S to P more easy. 9.12.3 Nature of enzyme action. Each enzyme has a substrate binding site in its molecule so that a highly reactive enzyme substrate complex is produced. This complex is short lived and dissociates into products. P and the unchanged enzyme with an intermediate formation of this enzyme product complex EP. The formation of ES complex is essential for the catalysts. The catalytic cycle of an enzyme action can be described in the following step. First, the substrate binds to the active site of enzyme, fitting to the active site. Second, the bindings of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. Third, the active site of the enzyme now is in close proximity of the substrate, breaks the chemical bonds of the substrate, and the new enzyme product complex is formed. Fourth, the enzyme releases the products of reaction and free enzyme is ready to bind to another molecule of substrate and run through the catalytic cycle once again. 9.12.4 Factors affecting enzyme activity. The activity of an enzyme can be affected by a change in the conditions which can alter the tertiary structure of the protein. These include temperature, pH, change in substrate concentration, or binding of specific chemicals that regulate its activity. Temperature and pH. The enzymes generally function in a narrow range of temperature and pH. Each enzyme shows its highest activity at a particular temperature and pH, called the optimum temperature and optimum pH. Activity declines both below and above the optimum value. Low temperature preserves the enzyme in a temporarily inactive state, whereas the high temperature destroys enzymatic activity because proteins are denatured by heat. Concentration of substrate. With the increase in substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity, Vmax, which is not exceeded by any further rise in concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzyme molecules to bind with the additional substrate molecule. The activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of the chemical shuts off enzyme activity, the process is called inhibition and chemical is called inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is called competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. मम्मी यार फोन ही करते रहते हैं. Consequently, the substrate cannot bind. As a result, the enzyme action declines. Example, inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure. Such competitive inhibitors are often used in control of bacterial pathogens. 9.12.5 Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on this type of the catalyze reactions. Enzymes are divided into 6 classes, each with 4 to 13 subclasses and named accordingly by 4-digit number. Oxidoreductase dehydrogenases, enzyme which catalyze oxidoreduction between two substrates. Transferases, enzyme catalyzing a transfer of a group other than hydrogen between a pair of substrate and S. Hydrolysis, enzyme catalyzing hydrolysis of ester, ether, peptide, glycosidic, C halide or P, phosphate and nitrogen bonds. Lyases, enzyme that catalyze removal of groups from substrate by mechanism other than hydrolysis, leaving double bonds. Isomerases, including all enzymes catalyzing interconversion of optical geometric or positional isomers. Ligases, enzyme catalyzing the linking together of two compounds. Example, enzyme which catalyze joining of carbon oxygen, carbon sulfur, carbon nitrogen, phosphate oxygen, etc. bonds. 9.12.6 Cofactors. Enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which a non-protein constituents called cofactors are bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzyme is called the apoenzyme. 3 kinds of cofactor may be identified: prosthetic groups, coenzymes, and metallines. Prosthetic groups are organic compounds and are also distinguished from other cofactors in that they are tightly bound to apoenzyme. For example, in peroxidase catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen, heme is the prosthetic group and it is the part of active sites of the enzyme. Coenzymes are also organic compounds, but the association with the apozyme is also transient, usually occurring during the course of catalysis. Furthermore, coenzymes serve as cofactor in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins. Example, coenzyme nicotinamide, adenine dinucleotide, that is NAD, and NADP contain the vitamin niacin. A number of enzymes require metal ions for their activity, which form coordination bonds with side chain as the active sites and at the same time. From one or the more coordination bonds with the substrate, example zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in catalytic activity of the enzyme.